Hey friends, my name is Aarti Sharma and welcome again to my YouTube channel. In this video tutorial, I'm going to talk about gene regulation in prokaryotes by using example of operon. So we are going to study about operon. Let's get started. So first of all, what is operon? Operon, it's a simply cluster of genes regulated by single regulatory control system that is known as operon. Here we have three genes which are going to be regulated by single control system, that is single promoter and single operator, that is known as operon. And these cluster of genes are known as structural genes which encodes for particular protein, and this promoter and operator region is known as regulatory region of the gene, right? In case of prokaryotic organisms, transcription and translation occur hand by hand. Means these organisms are the simple organism. They don't have any nucleus like eukaryotic organisms. So when their transcription occur, translation begins to occur simultaneously. Here this is the RNA polymerase enzyme. It is transcribing DNA into RNA. While it's transcribing DNA into RNA molecules, ribosomes simultaneously come and occupy mRNA and they begin to translate this mRNA into protein molecules, right? So here we can see that how simple prokaryotic organisms are, so that their transcription and translation is occurring simultaneously. Okay. Next, operon. Operon is also known as lactose operon. If we talk about the simple organisms that are bacteria, bacteria basically prefer to use glucose as a source of energy because glucose is a monosaccharide. It is easy to break down. But what happens when glucose is not present into the medium? Then in that case, if you provide lactose to the organisms in the medium, they will begin to utilize lactose in the absence of glucose. For utilization of lactose, they will require operon to express, right? Because operon contains some important genes which are important to express for the utilization of lactose, right? So these are the structural genes which are required for utilization of lactose, and the genes are LAC-Z, LAC-Y and LAC-A, right? The next is the structural genes are, first is LAC-Z, that is known as beta-galactosidase. Beta-galactosidase is used to break down lactose, that is a disaccharide, into glucose and galactose. These are monosaccharides and these are the simple sugars which are utilized by the bacteria easily. LAC-Y, it encodes for lactose permease. This enzyme helps lactose to move inside bacterial cell that is present into the medium. Lactose permease forms like carrier protein in the membrane of bacteria. It helps the lactose present in the medium to move inside of the bacteria, right? Next is LAC-A, that encodes for trans-acetylase. This protein helps some toxic chemicals to move outside of the cell, right? The next is basic gene regulation. In this case, we are aware about the regulatory region and structural region of the operon. Now there is one more gene that is known as LAC-I. It's also known as repressor protein. As its name represents, it is a repressor protein. So it helps in repression of the operon, right? The protein is active in its tetrameric form, right? It has two DNA binding regions and two lactose binding region. The protein will act like repressor protein in the absence of the lactose. When the lactose will not present, then these regions will bind to operator region, and when this protein will bind to the operator, then RNA polymerase will not be able to bind promoter region, and in that case gene expression will not occur, right? But when lactose is present into the medium, lactose will come and bind in this region. In that case, there will occur conformational changes in DNA binding region, and it will not be able to bind on the operator. In that case, RNA polymerase will come and bind to the promoter and gene expression will be able to occur, right? The next is, this is a simple assumption, like when glucose is present, lactose is not present, then lac operon will not express. When glucose is present, lactose is also present, then lac operon will not express. Why? Because simple source of energy is present. Bacteria will prefer to utilize simple source of energy as compared to the disaccharide. When glucose is not present but lactose is present, in that case lac operon should express. Okay, when glucose and lactose both are not present, then lac operon should not to express. So this is simple assumptions that we have made on the basis of the previous points that we have studied in lac operon, right? Now next, here you can see that this is the E. coli. E. coli is happy. Why? Because it has simple source of energy, that is glucose. So in this case, what will happen there? Lactose is not present. This repressor protein will bind to the DNA binding regions on operator. It will not allow RNA polymerase to bind promoter. In that case, gene expression will not occur, and bacteria is happy because it is utilizing glucose, that is the simple source of energy. In this case, you can see that the bacteria does not have glucose to utilize, but it is happy. Why? Because it has lactose in the medium. So in that case, the repressor protein will remain busy in binding with lactose and operator region will be free. Here RNA polymerase will come and bind and it will begin the gene expression. In that case, lac Z, lac Y, lac A will make proteins that will be used up to utilize the lactose present into the medium that will enter into the bacterial cell. Okay, now experimentally it has been seen that when glucose is not present, when glucose is present, lactose is not present, gene expression will not occur. But when both are present, abortive transcripts are formed. Now what is the reason behind of these abortive transcripts? First of all, our lac operon is a leaky operon. Leaky operon means, here this is the promoter, here this is the operator. Leaky operon means the binding between RNA polymerase and promoter region is weak. The binding is not strong. So the operon needs some protein molecules to strengthen its binding between promoter and operator, and that protein is known as catabolite activator protein. This catabolite activator protein is present in dimeric form. It becomes active when it binds with two molecules of cyclic AMP. Now besides of our promoter region, we have a cap binding region. When this active protein binds to the cap binding region, it helps to strengthen the bonding between RNA polymerase and promoter. Okay, but this process will happen only one condition. The process will happen when glucose is not present. Why? Because there is, okay, I will explain it in the next slide. Because there is inverse relationship between glucose and cyclic AMP. When glucose concentration is high, cyclic AMP concentration will be very low. In that case, the binding between RNA polymerase and promoter will be weak. Due to the low concentration of cyclic AMP, catabolite activator protein will remain inactive, right? And in that case, gene expression would be nil. When the concentration of glucose is very low, concentration of cyclic AMP will be high. Cyclic AMP will bind to the dimeric cap protein, and that will help to strengthen the binding between RNA polymerase and promoter region. In that case, gene expression will occur. Okay, so here you have seen that when glucose is present, cyclic AMP concentration will be very low. Lactose is also present. Okay, in that case, what will happen? The lac operon is a leaky operon, right? Here is promoter, here is operator. Due to weak binding, when RNA polymerase bind to promoter without any cap protein, Okay, without any active cap protein, there will not be strong binding between promoter and enzyme. This enzyme will bind and it will move. It will begin to express the gene, but due to weak binding, it will fall off early. Okay, and it will form abortive transcripts. Is it clear? Right? Due to weak bonding between promoter and RNA polymerase, the abortive transcripts will be formed in the absence of cap binding protein, because cap binding protein is important to strengthen the binding between RNA polymerase and promoter. So here I have written the reason behind abortive transcripts. Okay, next is, here you can see that here I have shown the region besides of promoter, that is cap binding protein, right? When glucose is present and lactose is not present, then what will happen? Glucose is present and cyclic AMP will be very low, and in that case what will happen? Almost nil gene expression. When glucose is not present but lactose is present, then cyclic AMP will be high. Catabolite activator protein is a protein which is almost present in the bacterial cell. When it will bind to the cyclic AMP, it will become active, and active catabolite activator protein will bind to the cap region, and it will strengthen the binding between RNA polymerase and promoter, and it will help gene to express, right? Next is negatively inducible operon. Your lac operon is negatively inducible operon. So what is the inducible? Inducible means operon require inducer to become activate, and that inducer is your allolactose, right? Lactose automatically convert into allolactose that act like inducer. So when this allolactose bind to the repressor protein, then this repressor protein will not bind to the operator. In that case, RNA polymerase will be able to bind to your promoter and gene expression will occur. Okay. Now why we call it negatively? Because when repressor protein is present in the absence of lactose, in the absence of allolactose, that is inducer, it will not allow gene to express. But when repressor protein is present in the presence of allolactose, then it will allow gene to express. Why we call it negative operon? Because the presence of repressor protein inhibit gene expression, while absence of repressor protein will not inhibit gene expression. That's why we call it negatively inducible operon system, right? So here is all about the operon. I hope you understand my lecture, and please like, share and like my channel. Thank you so much, friends. Thank you.